Hello and very warm welcome to everyone to our today's webinar on managing the challenging bioanalysis for PKPD assessment for new chemical entities. Hi, I am Amit Modi and I am president of the Vida Clinical Research. Vida Clinical Research Limited, together with its subsidiary Bioneeds India Private Limited and its joint venture Ingenuity Bioscience Private Limited, together referred as a Vida Group, offer a comprehensive portfolio of clinical, preclinical, and bioanalytical service to support the innovator, biosimilar, and generic drug development program of our global climate. Together, we serve the client globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal and nutrachemicals, and medical devices industry. As your facilitator today, I would like to introduce you to all the couple of points as we begin this session. This session will have a presentation in the beginning followed by the Q&A round. We have enabled our ask question features in the right on your screen where you can drop all your question related to this webinar right there. Now moving forward, I would like to introduce you all to our speaker and panelists for the today's session. First of all, I like to introduce our speaker today, Miss Ekta Bharwada. Miss Ekta Bharwada is working as a group leader at Vida Clinical Research Private Limited, Ahmedabad, India, where her role is to serve the scientific and technical lead for a biopharmaceutical companies. Ekta has a over 8+ year experience, professional experience, research in research. Also, she is involved in a regulated bioanalysis in developing, validating, and applying bioanalytical method to support the clinical study of generic molecule and new chemical entities. She plays a key role in the development, validation, and application of bioanalytical assay to comply with scientific and regulatory requirements. Now I can move forward to our panelists. We have today 3 panelists. The first of all, I would like to introduce our Miss Swati Gutikar. Mrs. Swati Gutikar is a head of bioanalytical, also the vice president of at Vida Clinical Research Private Limited, Ahmedabad, India. She has a more than 30+ years of professional experience in bioanalytical research, and her role is to bioanalytical scientist in a method development and method validation activities. And also she supervises the activities performed in regulated bioanalysis. She has published several research papers and articles in peer-reviewed scientific international journal like Bioanalysis, Journal of Chromatography, Journal of Chromatographic Science, Journal of Pharmaceutical Analysis, and Bioanalytical Chromatography, etc. Now I can move forward to our another panelist, Dr. Chirag Patel. Dr. Chirag Patel is working as a bioanalytical investigator at Vida Clinical Research Private Limited, Ahmedabad, India. He has a more than 17+ years of professional experience in bioanalytical research. His role is to guide the bioanalytical scientists in method development and method validation activity and supervise the all involved activity as per the regulated bioanalysis. He has published several research article paper in international journal, also in the Journal of Chromatography as well. Now move forward to our last panelist, Dr. Vikas Trivedi. Dr. Vikas Trivedi is working as a bioanalytical investigator at Vida Clinical Research Private Limited, Ahmedabad, India, where his role is served to scientific and technical aspect to pharmaceutical company. Vikas Trivedi has over 17+ years of professional experience in research and development, validating a very difficult method. Also, he has a professional experiences in developing a method for new chemical entity. He also play a key role in the developing, validation, and application of bioanalytical assay to comply with scientific and regulatory requirements. Vikas Trivedi has a overall 17+ years of experience in this field. So without further any delay, I would like to virtually call upon the Miss Ekta Bharwada, who is our speaker of this session today. Over to Ekta.
Okay. Thank you, Mr. Amit, for such a nice introduction. Here I welcome you all for today's webinar, Managing Challenging Bioanalysis for PKPD Assessment for New Chemical Entities. So today we will going to discuss about following points, which will cover background of NCE, PKPD and biomarker assessment of NCE, role of central laboratory for NCE, selection of CRO and diagnostic lab for NCE, then several challenges which can be faced in NCE studies and its management. Then followed by study sample analysis in NCE, and finally the expertise of VIDA in NCE. So first of all, we will see what is NCE and what is preclinical study and clinical study in NCE. So a new chemical entity is a term used to describe a compound that has never been approved for use in humans before and is not structurally similar to any previously approved drugs. So NCEs are usually developed through a process of drug discovery, which involves identifying and synthesizing new compounds that have the potential to treat a specific disease or condition. So once an NCE has been identified, it undergoes preclinical testing to evaluate its safety and efficacy in animal models. After NCE shows promising result in preclinical studies, it may proceed to clinical trials in humans to further evaluate its safety and effectiveness. Once NCE, so now regulatory submission. Once NCE successfully completes clinical trials and is found to be safe and effective, data will be submitted to regulatory agencies for further use in the general population. Next is next. So now we will see basic key requirements for NCE studies. So when we are going for NCEs, some key requirements are there, like study design. The study design should be robust and meet regulatory requirements. It should be planned in such a way that it can answer the research question and objectives of the study. Next is ethical considerations. So NCE studies involve human subjects and must adhere to ethical guidelines. All studies must be approved by an independent ethics committee and informed consent must be obtained from all participants. Then next is site selection. Sites for the clinical trials must be selected based on various factors, including the availability of suitable patient populations, expertise of the investigators, and regulatory requirements. Followed by recruitment and retention. So recruitment of participants can be a challenge in NCE studies. So appropriate strategies must be in place to identify and recruit eligible participants. Then data management. So the data generated during the study must be collected, managed, and analyzed appropriately. The data management plan must ensure the integrity, accuracy, and confidentiality of the data. Regulatory compliance. All NCE studies must be designed and conducted in accordance with GCP guidelines and other applicable regulations. Then followed by monitoring and reporting. So NCE studies must be monitored to ensure that they are conducted in accordance with the protocol and regulatory requirements. The study report must include all the required information as per regulatory guidelines. Next is, now we will see the PK assessment for NCE. So what is pharmacokinetic assessment? Pharmacokinetic assessment is a crucial step in the development of new chemical entities as it provides essential information about the drug's behavior in the body, in which preclinical studies are conducted in animals to determine the PK properties of the drug, including its absorption, distribution, metabolism, and excretion. These studies may involve single dose or multi-dose administration of the drug and help to identify the most appropriate animal species for further studies. Then clinical studies. So once safety and preliminary efficacy have been established in preclinical studies, clinical trials are conducted to assess the PK properties of the drug in humans. So here PK modeling and simulation can be used to predict the PK properties of the drug in different patient populations and to optimize dosing regimens. Next. So further clinical study includes 4 phases in which Mr. Amit, go to previous slide, please. Okay. So in PK assessment, clinical study includes 4 phases in which phase 1 studies typically involve 20 to 80 healthy volunteers and are designed to assess the safety and tolerability of the drug. Phase 2 and phase 3 studies are conducted in patients to assess the drug's efficacy and safety in treating the target disease. In that, phase 2 studies involve patients around 200 to 800 and phase 3 studies involve patients around 1000 to 5000. In phase 4, the safety of drug will be evaluated once it is reached to the market after regulatory approval. The primary purpose for conducting post-marketing surveillance is to identify previously unrecognized adverse effects as well as positive effects. Next. Now we will see PD assessment for NCE. So pharmacodynamic assessment provides information about the drug's mechanism of action, potency, safety, and tolerability. PD assessment typically involves studying the drug's effect on the target receptor, pathway, or physiological system in preclinical and clinical studies. So here also PD modeling and simulation can be used to predict the drug's effect on different patient populations and to optimize dosing regimens. Next. Now we will see the role of biomarkers in NCE. So biomarkers play an important role in the development of NCEs by providing valuable information about a drug's effectiveness, safety, and mechanism of action. Biomarkers can be used to provide early indications of a drug's effectiveness and safety. By measuring specific biomarkers in patients receiving the NCE, researchers can gain insights into how the drug is affecting the body and whether it is having the desired therapeutic effect. So there are some biomarker tests which can be helpful for NCEs like genetic biomarker test, protein biomarker test, imaging biomarker test, pharmacodynamic biomarker test, and safety biomarker test. Next. Now we will see the role of central laboratory in NCE. So what is central laboratory? The central laboratory is typically responsible for a range of activities, including sample analysis, data management, and quality control. During NCE development, the central laboratory plays a critical role in conducting clinical trials and ensuring that the drug meets regulatory requirements for safety and efficacy. So some of the specific tasks which can be performed by a central laboratory are like collecting and processing bioanalytical samples for clinical trial participants. Then managing and storing large volumes of data generated during clinical trials. Then analyzing samples using various techniques such as liquid chromatography, mass spectrometry, and immunoassays. Next is conducting quality control checks to ensure that data and sample analysis are accurate and reliable. Then interpreting the results of sample analysis to assess the safety and efficacy of the NCE. And finally, reporting data to regulatory agencies as required for drug approval. Next. Now we will see the diagnostic lab selection for NCE. So selecting the right diagnostic laboratory is most critical part for the success of NCE molecule development. When selecting a diagnostic laboratory, there are several factors need to be considered, like accreditation. So diagnostic laboratories should be accredited by an appropriate regulatory agency or third-party organization, such as the Clinical Laboratory Improvement Amendments or the College of American Pathologists. Next is expertise. The diagnostic laboratory should have a team of highly qualified and experienced professionals with expertise in the specific diagnostic test required for the NCE molecule. Next is capabilities. The diagnostic laboratory should have the necessary equipment and infrastructure to perform the required diagnostic test accurately and efficiently. Then data management. The diagnostic laboratory should have robust data management systems to ensure the integrity, confidentiality, and accuracy of data generated. Followed by regulatory compliance. The diagnostic laboratory should have a strong understanding of regulatory requirements, and it should be able to meet all relevant regulatory requirements. Then cost and location. So it is most important to select a diagnostic laboratory that offers competitive pricing and still maintaining high quality standards, and it should be near to clinical trial sites. Next. Now we will see the selection of CRO for NCE. The selection of CRO for NCE development is a critical decision that can significantly impact the success of the drug development program. Some factors that should be considered when selecting a CRO are like, first is experience and expertise. So the CRO should have a team of experienced scientists who have expertise in the required fields, such as medicinal chemistry, drug metabolism, pharmacology, toxicology, and formulation development. Then regulatory compliance. The CRO should be compliant with the regulatory requirements of the countries where the clinical trials will be conducted and should have a good track record of regulatory compliance, including successful inspections by regulatory agencies. Next is quality assurance. So the CRO should have a robust quality assurance program to ensure that the studies are conducted according to GLP and GCP guidelines. Then infrastructure and cost. The CRO should have state-of-the-art infrastructure in terms of well-equipped laboratories, animal facilities, and clinical research centers, and it should provide transparent pricing and have a good reputation for delivering services within budget and timelines. Then references. So before finalizing any CRO, it is essential to check their references. This includes speaking to previous clients to understand their experiences with the CRO and their satisfaction with the quality of work delivered. Next. So now we will see the several challenges which can be faced during NCE molecule handling. So first, we will going for challenges in NCE molecules handling. So handling NCE molecules can pose several challenges like toxicity. NCE molecules can be toxic to both humans and the environment. Therefore, their handling requires strict adherence to safety protocols and regulations. Next is solubility. NCE molecules may be poorly soluble in water or other solvents, making it challenging to dissolve and prepare them for further use. Next is stability. NCE molecules can be unstable and may undergo degradation, oxidation, or other chemical reactions over time, which can make it difficult to store and maintain their activity. Next is formulation. So sometimes NCE molecules may require specialized formulation techniques such as nanotechnology for liposome encapsulation to improve their bioavailability and efficacy. Then scale up. NCE molecules may be challenging to produce in large quantities, which require specialized equipment and expertise to manufacture them in bulk. Then intellectual property. NCE molecules may be subject to patent protection, making it challenging for other companies to develop similar compounds. Next. Now we will see the challenges faced by sponsor for NCE handling. So since NCE developments are complex and challenging, there are several key challenges that sponsors may face, like regulatory compliance. Sponsors must ensure that their NCE studies comply with FDA or other regulatory requirement, which can be complex and time consuming. Then next is funding. So conducting a study for NCE can be expensive and sponsors may face challenges in securing funding for the study. So this can be managed by seeking funds from investors or applying for grants or loans. Then recruitment of participants. Finding suitable participants for NCE studies can be challenging, especially if the study requires specific patient population or if the study is conducted across multiple sites. So recruitment efforts may involve working with patient advocacy groups or collaborating with other research institutions. Then study design. So sponsors must carefully consider the study design, including the choice of endpoints, control groups, and statistical analysis to ensure that the study meets its objectives and produces meaningful results. The next is data collection and analysis. Sponsor must ensure that the data is collected accurately and analyzed appropriately. This may involve working with specialized data analysis software or contracting with outside data analysis firms. Then safety and ethical concerns. So NCE studies must be conducted with the participants' safety and ethical considerations. Sponsors must ensure that the studies protocols protect participants' privacy and confidentiality, and that if any of the adverse events are there, it should be reported appropriately. Next. So now we will see the challenges faced by CRO for NCE analysis. So NCE is a complex, there are some challenging tasks which can be faced by CRO during the analysis of NCE samples, which we will discuss one by one. First is limited sample availability. The availability of samples can be limited due to various reasons, such as the high cost of synthesis, the limited amount of the compound available, or the need to maintain the confidentiality of the specific compound. Then next is complexity of the molecule. So NCEs are often structurally complex molecules that require sophisticated analytical techniques for their analysis. The complexity of these molecules can make it difficult to isolate and characterize their different components accurately. Next is sensitivity and selectivity of analytical techniques. The analytical techniques for NCEs should be highly sensitive and selective to detect and quantify trace amount of these compounds accurately. To achieve this parameter requires highly skilled analysts and state-of-the-art analytical instruments. The next is lack of reference standards. The absence of reference standards can make it challenging to determine the purity, potency, and identity of these compounds accurately. So sponsor needs to provide valid standards to CRO for NCE analysis. Next is regulatory compliance. So CROs need to comply with various guidelines and regulations related to sample handling, storage, and analysis. Then turnaround time. So CROs are often required to provide fast turnaround times for the analysis of NCE samples. So achieving this can be challenging, especially when dealing with complex samples that require multiple analyses to obtain accurate results. Last is cost. The analysis of NCE samples can be expensive due to the need for highly skilled analysts, state-of-the-art analytical instruments, and specialized reagents and materials. Next. Now here we will see the specific challenges which can be faced during the bioanalysis of NCEs. So first is method development. Method development for NCEs should be done by taking all available earlier information from preclinical study and available literature of the molecule. If there is any expected metabolite and it is feasible, it should be the part of the development. Then next is dynamic linearity range. So linearity range of methods should have minimum dose level PK to multiple ascending dose levels to cover a large dynamic range. In case of any further higher concentrations, it requires pre-dilution of the samples. Then sample volume for analysis. Due to limited blood collection and multiple determination requirements, limited aliquot volume is recommended during method development. Therefore, in some cases, there are new techniques such as capillary, DBS cards, or tape device can be used. Next is metabolite determination. As a well-characterized metabolite may not available in early trial, so exploratory method may require to separate the expected metabolite and determine the level of concentration. Next is method reproducibility. So as the trial goes long duration, methods should be rugged and reproducible in terms of lab variations. Sometimes there is need to involve multiple labs in same trial, so cross-validation is mandatory to present the data reliability between the labs. The next is long-term metric stability. So for NCE molecule, as the trial continues to years, long-term stability approx 3 to 5 years should be required. Next is method activeness frequency. So methods should be active and frequently tested for performance to avoid multiple repeats due to criticality of the samples. Next. Now we will see several method development challenges for NCE. So during method development of NCE, there are many challenges that scientists can face, which includes ensuring target selectivity. So NCEs must be designed to selectively interact with a specific target or set of targets. However, achieving target selectivity can be challenging as many targets have similar structures or functions. So developing methods to test for target selectivity and minimize off-target effects is crucial to the success of NCE development. Next is optimizing pharmacokinetics. NCE study must have appropriate pharmacokinetic properties, such as adequate bioavailability, half-life, and clearance. So developing methods to optimize pharmacokinetic properties, such as in vitro and in vivo assays, is critical for evaluating the efficacy of NCEs. Then characterizing the drug metabolism and toxicity. NCEs must be screened for their metabolic stability and potential toxicity to ensure that they are safe for human use. So developing methods to test for drug metabolism and toxicity, such as in vitro assays and animal models, is crucial for the evaluating the safety of NCEs. Then overcoming the solubility and formulation issues. As we have discussed, NCEs may have poor solubility or stability, which can limit their usefulness as pharmaceuticals. So methods should be developed by using solubilizing agents or nanoparticle formulations. Is important for improving the pharmacokinetic and efficacy of NCEs. Then meeting regulatory requirements. So developing NCEs that meet regulatory requirements is a complex and challenging process. So regulatory agencies require extensive preclinical and clinical data to be submitted before approving a new drug for use in the humans. Next. So now here we will see logistics and timeline challenges in bioanalysis of NCEs. First is sample management. So there should be a coordination between sites, custom clearance, and logistic is required for import of the samples. Then reference standard management. The application of import license and its approval is required for process of custom clearance and logistics. Next is sample results timelines. So first in human study, the timelines are critical to share PK data along with safety profile review for further decision on trials. So from sample receipt to analysis, result sharing duration is 3 to 4 days. And in case samples are more, then multiple equipments can be included to meet the timelines. Then faster switching and decision making. So sometimes the predicted range doesn't work for the analysis. So real faster switching is required to update and revalidate the method. Next is communication. Faster communication is key for any successful trial. So PK scientists should be timely revert the trial leads for required information. For safety cases, some communications are critical for trials which affect further progress and decision. Then data flow. The generated PK data should be reliable and acceptable as per global quality standards. So if any mistakes are there in data, should be communicated appropriately with the sponsor. Next. Now we will see the flow of study sample analysis for NCEs. So there are several points which need to be considered during study sample analysis of NCE. Like before start of the study sample analysis, reference or working standards must be procured in advance along with other consumables. Then sponsor specific requirements should be included in bioanalytical study plan, which includes method specific information, clinical protocol information, analysis information, and stability information. Then training of bioanalytical study plan, method SOP, and clinical protocol should be conducted prior to start of the study sample analysis. After training, trained person will start study sample analysis, and based on initial analysis data, pre-dilution may be included if required. Next. Then dedicated custodian team is required for sample receipt and sample verification, along with Watson-Lims team person, and coordination between all the teams and sponsor must be required. Any sample discrepancy is there, it needs to be communicated immediately to the sponsor for further resolution. Then after completion of analysis, data should be reviewed properly and further results should be transferred to sponsor or third party through digital interface to avoid any manual error. Further results should be reported in study report properly with all information, and data must be archived as per archive duration. Next. Now we will see some important parameters which can be included in study sample analysis. An analytical run, which is consist of a blank sample, a zero sample, calibration standards, at least 3 levels of QCs in duplicate and the study samples to be analyzed. The QC should be interspersed in the run in such a way that the accuracy and precision of the whole run is ensured. Then acceptance criteria or rejection of the analytical run should be defined in the protocol, in the study plan, or in SOP. When a run contains multiple batches, acceptance criteria should be applied to the whole run and to the individual batches. Then possible reasons for reanalysis of study samples, the number of replicates, and the decision criteria to select the value to be reported should be predefined in the protocol, study plan, or SOP. Reinjection of processed samples can be made in the case of equipment failure if reinjection reproducibility has been proven during method validation. Then chromatogram integration and reintegration. It should be described in a study plan, protocol, or SOP. Then incurred sample reanalysis should be carried out to verify the reliability of the reported sample analyte concentrations and performance of the method. And next is cross-validation. So cross-validation is required to demonstrate how the reported data are related when multiple bioanalytical methods and multiple bioanalytical laboratories are involved. Next. So now we will see the expertise of VIDA in NCE. VIDA has developed and validated 1151 method till date, and among these, 83 methods are of new chemical entities. VIDA has developed methods for endogenous molecules, hormones, steroids, inhalation formulations, chiral and liposomal products, etc. VIDA has average processing capacity of 1 lakh samples per month, and VIDA worked as a central bioanalytical laboratory for global phase 2 and phase 3 trials. So here I'm ending my session. Thank you.
Thank you, Ekta, for wonderful presentation. And it gives a simple description of this presentation for this topic. So now moving to, we'll be proceeding ahead with our discussion, where we are going to answer some of the interesting question posted by our audience. So first of all, we received the one question from the audience is, what is the current regulatory requirement to import the sample, and how much time it is generated to require this document? I think I'll ask this question to Dr. Chirag. Can you please explain what is the requirement and procedure for that, and how much time it will require to import this sample? Over to Dr. Chirag.
Yeah, thank you, Amit. And regarding the import of biological samples in India, at present there is no import permit is required to import the samples in India. Shipment can be shipped through courier agency, that's a professional or logistic agency, and the documents which is required while shipping the samples from out of India, and the documents which is required for the custom clearance are commercial invoice, then the packaging list, then the non-hazard letter, undertaking of biological samples, BRC declaration, and use letter, and the declaration letter. With the proper coordination between the shipper, consignee, and the courier agency, shipment of samples can be delivered to India within the 7 to 10 working days. Over to you, Amit.
Thank you, Dr. Chirag, for explaining each and every aspect for importing the sample. Now we have another question from our audience is, how PK sample are planned to analyze, if it will be in batches or it will be an individual sample analysis, and what is the impact if we analyze the sample in individual batches from each subject? Can I, Dr. Vikas Trivedi, could you answer this question, please?
Yeah, thank you, Amit, for giving me the opportunity to address this question. Specifically, planning for the analysis of PK sample for new chemical entity require proper planning, as these type of samples always comes in a cohort and in a different time interval manner. Furthermore, the results of these samples must be delivered in short duration of time after receiving the samples, because specifically for the first in human studies, the sponsor or we need a results on the priority basis. Therefore, biological lab must be ready to analyze of these samples in a quickly manner, or all the initial activities should be planned in well in advance. In addition to these, batch-to-batch pharmacokinetic variability confirm the current regulation, which create the question for the reliability of the trials. So to overcome this type of challenges or this type of situation, biological lab must be take care of certain steps, such as bulk spike samples should be used throughout the study sample analysis, ensure the suitability of working and reference standard before it usage. If feasible, then same equipment needs to be used, and also the in-cut sample reanalysis should be performed in parts by selecting the samples from the multiple batches to ensure the reliability of the results. Thank you. Thank you, Amit. Over to you.
Thank you, Dr. Vikas, for a nice explaining. So we have received the another question is, as which is the most important factor for selection of the CRO? As we know that there are many CROs available in pharmaceutical industry. So considering this point for the selection of CRO, which is the most important factor should sponsor consider for the selection of CRO? And can Ms. Swati Guttikar, please, ma'am, can you explain regarding this?
Yeah, thank you, Amit. As described by Ekta, she has already given the factors which are considered for selecting of the CRO. So to summarize here, the selection of CRO is based on the experience and expertise of the CRO, the infrastructure, having the state-of-the-art lab with the sensitive equipments, the competitive pricing, the robust quality assurance team, the regulatory compliance, and the good regulatory track record. So in this, if we see, the experience and the expertise is the most important factor for the CRO, as the CRO needs to have a hands-on experience on handling the NCE projects. And the experienced CRO has the expertise in troubleshooting any challenges faced during the developments and validations. And the experienced CRO have a good exposure to the regulatory requirements of the documents for submissions to various regulatory agencies. Thank you, Amit.
Thank you, ma'am, for a nice explaining. And I would like to thank you all, the panelists, for nice answering all our questions and from the audience. So I would like to take a pause over here. We understand that this session has kept us all well engaged throughout, but due to time constraint, we would not able to take any further question at this time point. But please be rest assured that we will be sending the answer to every question of your via email. Also, we will be sending across the record link of this webinar on the all registered email ID. Lastly, and on behalf of the Vida Clinical Research, would we like to sincerely thank you for for the being here today and taking out the time to be a part of this webinar. I would like also to extend our gratitude to all panelists for being such a wonderful presentation and giving us all a great learning today. We at the Vida Clinical Research honestly look forward to hosting you all again. Till then, take care and safe for everyone.